guys, welcome to Dr. Science. So today we are going to see the structure of protein kinase A. So first we will draw a small diagram here in order to understand this. So this is the plasma membrane, and on the plasma membrane there is a receptor, and this receptor is passing 7 times through the plasma membrane. Hence this receptor is known as your 7-pass receptor. Not only that, on the plasma membrane we have 3 proteins. One is your alpha protein, one is your beta protein, and one is your gamma protein. Okay. So imagine there is a ligand. There is a ligand or hormone coming and getting attached to the 7-pass receptor. Whenever this happens, there will be a conformational change in the 7-pass receptor, and this will eventually go to the alpha subunit and stimulate the alpha subunit. Whenever the alpha subunit is stimulated, it will convert the GDP into GTP. Now this GTP will eventually go to a complex. This complex is known as your adenylyl cyclase. So the GTP will get converted into cyclic AMP with the help of your adenylyl cyclase. Now the cyclic AMP will eventually go to protein kinase A, which is an enzyme, and activates the protein kinase A. Eventually this protein kinase A will do phosphorylation of certain proteins. Okay. So today we are going to see the structure of this protein kinase A. So protein kinase A is a tetramer, and this now tetramer means it have 4 subunits. Out of these 4 subunits, 2 subunits are regulator units and the other 2 are your catalytic units. So we will try to draw the diagram here. So it contains 2 regulatory units, and not only that, it contains 2 catalytic units which are linked together. Now this entire complex is your protein kinase A, but it is in inactive form. Okay, this protein kinase A is in inactive form. Now imagine there is activation. Activation is by cyclic AMP. Now there is this activation of protein kinase A by cyclic AMP. Now can you see this R subunit? These are the regulatory subunits. These regulatory subunits are literally doing repression, or these proteins are your repressors. They are repressing the activity of the catalytic unit. So we have to remove the repressor subunits in order to activate the protein kinase. So the repressor proteins will get separated from the catalytic units. Now these catalytic units are nothing but your active form of your protein kinase A. Now these catalytic units or the active form of protein kinase A will do phosphorylation of proteins, especially at your serine or threonine amino acids. Now what is meant by this? As the name suggests, protein, let me draw this clearly. So protein kinase. Kinase means an enzyme. Whenever you hear the word kinase, it means it is an enzyme which will do phosphorylation. An enzyme which will do phosphorylation or add phosphates is known as your kinase enzyme. So this kinase is going to add phosphates on a protein. So proteins are nothing but amino acids, right? So here is amino acids. and all these amino acids fuse with each other, and the entire complex is nothing but your protein. So amino acids fuse with each other and they will form a long chain which are called as your proteins. So I already told you protein kinase will do phosphorylation at serine or threonine amino acids. Now what is meant by this? So here is an amino acid, here is an amino acid, here is an amino acid. Now there is an amino acid named serine. Okay, so protein kinase will do phosphorylation at the serine amino acid. Similarly, here is an amino acid, here is amino acid, here is amino acid, and here is an amino acid named threonine. So here also the protein kinase will do phosphorylation of the protein. So this entire thing is your protein. Now there are certain enzymes which can break this phosphate. Those enzymes which can break these phosphates are called as your phosphatases. Okay, and not only that, I already told you that whenever there is receptor activation, alpha subunit will convert the GDP into GTP, and GTP will eventually get converted into cyclic AMP by the help of adenylyl cyclase. Now this cyclic AMP will have a cyclic AMP will induce protein kinase. Protein kinase will do phosphorylation of certain proteins, and there will be a physiological response. So, for example, you can think you are doing exercise, so you need a lot of energy. So this cyclic increase in cyclic AMP will lead to a physiological response such as doing exercise. Now this cyclic AMP can be broken down into 5' adenosine monophosphate. If that happens, there will be decrease in the cyclic AMP, right? So there will be decrease in the physiological response. So the enzyme which is converting this cyclic AMP into 5' AMP is your phosphodiesterase. Okay, so phosphodiesterase is an enzyme, which converts cyclic AMP and inactivates the cyclic AMP into 5' AMP. Okay, now why this is important? Because there are certain things like caffeine, and this caffeine is a phosphodiesterase inhibitor. Now what happens if there is a phosphodiesterase inhibitor such as your caffeine? So obviously there will be cyclic AMP, and cyclic AMP will be eventually gets inactivated into 5' AMP or adenosine monophosphate. Now this was done by phosphodiesterase. Now we are going to inhibit this phosphodiesterase enzyme by adding caffeine to these people. So what will happen? There will be increase in cyclic AMP, increase in protein kinase activity, so increase in physiological response. For example, you will not sleep whenever you are taking caffeine because there is increased cyclic AMP. Okay, so that was about the structure of protein kinase and phosphodiesterase. So before ending the topic, this is taken from your test book. As you can see here, this ATP or GTP gets converted into cyclic AMP by adenylyl cyclase, and eventually the phosphodiesterase can decrease the cyclic AMP by converting into 5' adenosine monophosphate. Now as you can see here, cyclic AMP will go and convert the inactive protein kinase, which have 2 regulatory subunits and 2 catalytic subunits, into the active form of protein kinase, which have the catalytic subunit. As you can see here, the repressor protein is getting ejected out. Okay, during this step, the protein kinase is doing phosphorylation of protein. So this protein This protein will eventually get a phosphate on its serine or threonine residues. So as you can see here, then it is called as a phosphoprotein. Now these phosphates can be broken by an enzyme called as phosphatase and convert the phosphoprotein into a protein, and it can have the physiological effects. So that was about protein kinase. Thank you guys.